Over the years, Irish people have used their own land to nourish themselves and their families. In much the same way, Irish investigators are looking to certain crops to see if they can harvest their inherent health-giving properties.
The focus of my research really is to try and increase levels of compounds that are already present in the food that have beneficial properties. So just enhancing the levels of beneficial compounds in the food itself.
Dr. Nigel Brunton is looking at the health-giving properties which naturally occur in certain foods. One of those compounds that interests Brunton is beta-glucan.
Beta-glucan is basically an insoluble fibre. It consists of a long chain of sugars. And the reason why we're very interested in beta-glucan is that it has been shown to have a lot of properties beyond basic nutrition. For example, it can promote immune function, it can have a probiotic effect. It can also lower cholesterol, and that's the reason why we're actually trying to enhance levels of beta-glucan in a bread. We already knew from other people's results that barley had high levels of beta-glucan in it. So the first step into producing a bread is to actually mill the barley grain. During the milling process, you generate a number of different fractions. For example, you produce a flour, you produce a bran. What we were very interested in is to look at the levels of beta-glucans in these different fractions.
Brunton's research threw up some interesting and unexpected results.
The exciting thing, I suppose, was that we found 4 times the level of beta-glucan in the barley end fraction, and this is the fraction that is normally thrown away, so therefore we're actually increasing the value of the barley while also increasing the nutritional properties of the breads that we were going to incorporate the barley ends into. So the next challenge then is to take that fraction and incorporate it into a bread. And what we want to do is to produce a bread that tastes as good as normal breads that are on the supermarket shelf, which has higher levels of this helper-making compound in it.
For normal white wheat bread, people are very used to its taste and used to its flavour. So when you're replacing wheat flour with something else, it's always quite difficult to try and get something that will simulate wheat bread completely. When you're making bread, you want to get a viscoelastic dough, a dough that you can stretch and knead into the form of a bread loaf shape. And it's the gluten in wheat flour that is responsible for this property, and therefore it's very difficult to try and get an elastic dough if you don't have the gluten present. So that's what we're trying to do at the moment. Okay, well, this is a normal wheat bread that you would find in the supermarkets. It is a 100% wheat flour formulation, and these two breads would have different levels of barley inclusions. This would have 15% of barley germ. This would have a much higher level of barley beta-glucan, and as you can see, there's an optimal level before the product quality will start to decrease. So what we're doing at the moment is we're trying to find the optimum level of barley inclusion without detrimentally affecting the quality of the breads. Well, I think that the functional food area is going to grow a lot. I think that bread, because it's such a universal product, it makes for a very easier carrier for healthy ingredients. Research is starting to increase at the moment now in the area of nutritious cereal products, so definitely in the future it's an area that's going to expand.
While scientists like Nigel Brunton continue to examine the naturally occurring health-giving properties of food, others are attempting not to exploit the inherent compounds of plants, but to use them as factories to grow foreign cells inside them. For centuries, scientists have been looking at plants for their healing properties. Today, investigators are looking at plants in a new way, which may prove helpful in the fight against a range of diseases. Phil Dix of NUI Maynooth is working to change the genetic makeup of key plant cells, results of which might play a critical role in human disease such as HIV/AIDS.
Well, plants have always been a very important source of medicines. There are many, many examples of a wide range of plants. And this is an example behind me, in fact, of a medicinal plant. This particular yew tree is a very famous one. It's probably more than 1,000 years old or thereabouts, possibly the oldest in Ireland. And it's known as the Silken Thomas yew because Reputedly, Silken Thomas, the 10th Earl of Kildare, played the harp under this on his last night in Maynooth before his rebellion was finally crushed by Henry VIII's forces. Well, in the 1960s, it was discovered that the bark of this tree contains a chemical called Taxol, which is very effective against a range of cancers. The chemical is produced in the bark. If you remove the bark, you kill the tree. And it's also produced in relatively small amounts, so you have to sacrifice a lot of trees in order to treat patients. Now, with the new genetic technologies available to us, we can start looking at very different medicines, ones that aren't necessarily of plant origin at all. For example, we could look at the production of human insulin, or antibodies, or vaccines. There's a possibility of using proteins produced in plants to help in the efforts to produce a vaccine against AIDS.
Phil Dix is looking at plant systems as a way of growing key proteins involved in the HIV disease mechanism. This is a revolutionary new system by which a plant is selected as a form of surrogate host, within which it's possible to grow a completely foreign compound, such as this key protein. Traditionally, the methods of producing these proteins were in bacterial or animal cell cultures. However, these are extremely expensive, and because they run a serious risk of contamination, they have to be maintained in highly sterile conditions.
Plants have a big advantage over these systems for several reasons. Firstly, you can grow up large amounts of plants, a large amount of material in a confined space very cheaply. In addition to that, there's no risk of infection with animal viruses that you can get in animal cell cultures, or the production of toxins that you associate with microbial cultures. We use tobacco for this work because it's a model plant for the plant tissue culture procedures. It works very well for these methods we have to employ. But in addition to that, it's obviously a plant which produces a lot of leaf material, which is where we're producing the protein of interest.
In HIV, the virus intercepts the human cell. Inside the virus, the RNA, which contains the viral genes, begins to replicate and infect the cell. The P24 protein surrounds and protects the RNA. With the replication complete, the virus then exits the cell and begins targeting other healthy cells. If antibodies can target the P24 protein and knock it out, it will destroy the virus's capability for protecting its harmful genes, thus exposing it to potential vaccines.
So the idea is to introduce the gene that's responsible for making the protein of interest, the HIV protein, into the plant cells so that we can produce the protein throughout the whole plant. And of course, if we then collect seed from that plant, we can grow up greenhouses full of those plants to produce the protein.
Dix's approach was novel. He bypassed the nucleus and targeted another site where the genetic material was plentiful, the chloroplast. The chloroplast has its own DNA molecules, up to 1,000 copies in the cell, as compared with just 2 in the nucleus. So when a foreign gene is introduced, it could be amplified up to 1,000 times, producing large amounts of the foreign P24 protein, making it a much more efficient production system.
Another attraction is that we can be very precise in where we insert the foreign gene into the DNA molecule that the chloroplasts have, in contrast to nuclear transformation, where that insertion takes place at random in the nucleus. This means we can avoid interfering with any other gene functions. In order to do this, we first of all grow some small sterile plants in tubs. We remove the leaves from these and place them in a special chamber in what we call a gene gun. This enables us to fire small particles of gold coated in our DNA, which includes our gene that we want to introduce. We fire those at the plant material. The DNA penetrates the plant cells, and some of it will end up in the chloroplasts. This can become incorporated into the chloroplast's genome, and gradually that new population of chloroplast DNA molecules can take over until all the DNA molecules within the chloroplast and then within the cell contain the gene of interest.
Having shown that they could grow the stable HIV protein in large amounts in plants, the next stage was to make sure that the protein was as effective in the new plants as it was in the bacterial models.
So we had actually a PhD student who came here for a few weeks and extracted the protein from the leaves, and she took the protein back to George's Hospital, and she's been injecting mice with an E. coli version of the protein and our plant-made version, and they found that the mice respond in the same way to both proteins, so our plant-made protein is as good as the E. coli protein.
Dick's and McCabe's work is attracting significant interest in this new and vibrant area of plant science. Researchers from around the world have gathered in Cork for a conference on this emerging field.
The work that Phil Dix is doing in Maynooth, targeting gene expression to the chloroplast for pharmaceutical production, is probably one of the leading labs in the world, because this is still pretty young science. We're just beginning to start looking at the notion of using plants for expressing human therapeutics, and there's really only a few leaders in the world. And I think Phil Dix is really in a position to be ahead of the curve, because he's one of the first to actually look at the notion of using chloroplasts for expressing human therapeutics. So Europe, and Ireland especially, I think could really make a name for themselves in this field.
I really believed and still believe that it's going to have enormous impact in the developing world. We're going to produce a variety of the new plant-based pharmaceuticals. It's going to benefit people in the developing world as well as, of course, the first world. But I think the big potential is taking high-tech medicines into low-tech parts of the world. These are the sorts of things I think that Ireland has been very good at adapting, taking new strategies for technology and implementing it in a high-tech way.
Well, we've had great success with the production of this particular HIV protein, P24, and we're hopeful that we'll be able to extend this to a broader range of diseases in the near future.
The use of plants as production systems for pharmaceuticals is gathering momentum throughout the world. It's clear that Irish investigators are delving deeper into the genetic and molecular makeup of crops and plants in a bid to better understand how to harvest the potential within. Although this is still a relatively young science, results to date indicate exciting possibilities that should bring us all significant benefits into the future.